Hi, good morning everyone. Today's topic is types of phosphorylation which occurs during photosynthesis and respiration in plants. First, what is phosphorylation? Phosphorylation refers to synthesis of ATP. How ATPs are synthesized during phosphorylation? There are three types of phosphorylation, but before learning phosphorylation, let us understand what is the meaning of oxidation and reduction. Reduction refers to addition of hydrogen, removal of electron. Example, during photosynthesis, you know that carbon dioxide is utilized. Utilized carbon dioxide undergoes reduction, which means to carbon dioxide, to that there will be addition of electrons and hydrogen. As a result, there will be formation of glucose. Hence, carbon dioxide is reduced during photosynthesis. Important point to remember. What is oxidation? Opposite of that. Losing hydrogen, gaining, losing electron. So both, if hydrogen, if the particular substrate is losing hydrogen, it will be losing electron also. That is oxidation. Example. In the first example, look at here. NAD plus, it is an coenzyme. NAD plus accepts electron and accepts one proton, two electron and one proton it accepts, turns into NAD plus was there. Now because of exception of H plus, it will turn into NADH. Hence, what has happened here, since it is accepting H, it is known as the reduction process. NAD, remember NADH will be rich in 2 electron and a proton. Now that NADH, if it loses its electron and proton, 2 electron which is gained and a proton which is gained, if it is losing it, then it is oxidation. NADH to NAD plus is oxidation, NAD to NADH is reduction. So that is about oxidation and reduction. While explaining phosphorylation, I will be explaining particular steps where there is reduction as well as oxidation. For that sake, I explained it. There are three types of phosphorylation: photophosphorylation, substrate level phosphorylation, and oxidative phosphorylation. What is photophosphorylation? Photo refers to photon. During photosynthesis, plants will be utilizing light. Light will be carrying packets of energy known as photons. That photon is used to prepare ATP. Let us understand. The photophosphorylation are of two types: non-cyclic photophosphorylation and cyclic photophosphorylation. Let us check non-cyclic photophosphorylation and understand how ATP is synthesized. Light is absorbed by the reaction center of photosystem 2, that is P680. The reaction center, when it absorbs light, its electrons gets excited. From where does electron come from? Electron come from splitting up of water. Water splits into 2 electron, 2 proton and half oxygen. Now you have to tell me which kind of reaction it is. Water is losing electron, losing proton. If it is losing, it is oxidation. So important thing, during photosynthesis, water undergoes oxidation. The lost electron from the water enters into reaction center of P680. In the presence of light, there will be excitation of electron. That excited electron is accepted by electron acceptor. Excited electron, now it will come back to ground state through series of proteins arranged, which forms electron transport system. I have written it as ETS. When electron is undergoing oxidation reduction in ETS, the energy is released. You have to remember that when there is continuous oxidation reduction in ETS, energy is released. That energy is utilized to combine ADP with inorganic phosphate. As a result, ATP molecule is formed. That is an example for photophosphorylation, formation of ATP. How this ATP got formed? It is because of light. Now what will happen? Electron will come to the reaction center of photosystem 1, that is P700. Again it is re-energized there. The 2 electrons which has come to the reaction center gets excited. It is captured by the electron acceptor. There in the electron acceptor, the 2 electrons are added to the one more compound known as one more coenzyme known as NADP. NADP will accept two electrons and the two protons which is released by splitting up of water turns into NADPH. This is what kind of reaction it is. It is accepting electron, two proton, hence it is a reduction reaction. So this is how ATP is synthesized by using light. That is first kind of phosphorylation known as photophosphorylation. Second type of phosphorylation is substrate level phosphorylation. What is substrate level phosphorylation? There will be a substrate, there will be an enzyme. What enzyme will do is, enzyme will be transferring the phosphate group directly to the ADP. Okay, so substrate level phosphorylation refer to enzyme transfers phosphate group directly to the ADP. Hence there will be preparation of ATP. Let us understand. During glycolysis at two places, at two steps, there is substrate level phosphorylation. These are important, you have to remember. First step, when there is conversion of 1,3-bisphosphoglycerate during glycolysis to 3-phosphoglycerate. What is 1,3-bisphosphoglycerate? In this particular compound, at the first position and the third position, 3-carbon compound, there is phosphate. When that first positioned phosphate is removed by removed in the presence of enzyme phosphoglycerate kinase, what phosphoglycerate kinase does is that phosphate which is removed is added to the ADP. That is the function of kinase enzyme. Anything related to ATP will be having kinase name. As a result, ADP, because of the addition of that phosphate, gets converted into ATP. So during glycolysis, in this particular steps, there is substrate level phosphorylation. Directly from substrate, phosphate is added to the ADP. That is substrate level. Second step, in the last step, where there is conversion of phosphoenol pyruvate to pyruvate. Where, in the presence of enzyme pyruvate kinase, again kinase related to ATP, where the kinase enzyme, phosphoenol will be having phosphate group. That phosphate is removed, added to the ADP. Again there is synthesis of ATP directly mediated by kinase enzyme. Hence pyruvate is prepared. So that is also substrate level phosphorylation. You understood? Directly from the substrate, phosphate is added to the ADP and ATP is synthesized. Next come to Krebs cycle. During Krebs cycle, there is conversion of succinyl CoA to succinate. Succinyl CoA will be having CoA group that will be having thioester bond. The thioester bond is broken. As a result, energy is released. Large amount of energy which is released can combine phosphate with the GDP. Why GDP? Because energy released is sufficient to combine GDP. As a result, GTP is formed. GTP again combines with ADP, transfers it, one of the phosphate group forms ATP. So this is also a substrate level phosphorylation because energy from the substrate is used. Succinyl CoA, as I mentioned, thioester bond. So that is about substrate level phosphorylation. Here at three places during cellular respiration, substrate level phosphorylation occurs. First two steps during glycolysis and the third step during Krebs cycle. Now in the first step, ATP, how many ATPs are released? As you know, the glycolysis, two pathways will be going on because there is splitting of 3-carbon in between. As a result, there is release of 2 ATP in this first step. In the second substrate level phosphorylation, this step also occurs twice, hence 2 ATP. So through glycolysis, during glycolysis through substrate level phosphorylation, 4 ATP, plus in Krebs cycle there is conversion of succinyl CoA to succinate. Two cycles will be going on because two pyruvates are prepared during glycolysis. Both of them will undergo Krebs cycle. Hence, one will release 1 ATP. Two reactions will release 2 ATP. Total 2 plus 2 plus 2, 6 ATPs are released during substrate level phosphorylation. Very, very important. During glycolysis, 4. During Krebs cycle, 2. That is about substrate level phosphorylation. Third one is oxidative phosphorylation. Already we learned what is oxidation. So oxidative phosphorylation refers to here there is involvement of NADH and FADH2. If NADH and FADH2, which is synthesized during cellular respiration, if it undergoes oxidation, there will be release of ATP. That is known as oxidative phosphorylation. So I have written when NADH and FADH undergoes oxidation, oxidation occurs in a series of protein complexes known as ETS, electron transport system, to form ATP, then it is oxidative phosphorylation. It is a big process, electron transport system. Let me concise it and explain it to you. You know that when there is entry of glucose into the cell, glucose will enter into the cytosol of the cell. It will undergo glycolysis. During glycolysis, glucose will break down into 6 carbon glucose will break down into 3 carbon 2 pyruvate molecules, during which there will be release of 2 NADH. That is one thing you have to remember. Second thing, pyruvate which is formed, 2 molecules, it will now enter into mitochondria. When it is entering into mitochondria, it will form acetyl CoA. During this step also there is formation of NADH. How NADH is formed? NADH is formed from NAD. NAD accepts electrons and protons, forms NADH. Hence you have to remember, all NADH which is formed, it is rich in electron, rich in proton. If that proton and electron is lost, there will be synthesis of ATP. That you remember. So acetyl CoA is formed. It is a connecting link. Acetyl CoA enters into Krebs cycle. There will be 2 Krebs cycle which will be occurring. So there will be release of through 2 Krebs cycles. There will be release of total 6 NADH and 2 FADH2. Again, FADH2, how it is formed? FAD will be there. FAD accepts proton as well as 2 proton and 2 electron. FAD, 2 proton, since it has accepted, it will become FADH2. So that's how 2 FADH2 will be formed. One cycle, 1 FADH2, 2 cycle, 2 FADH2 will be formed. Now all these NADH, 1, 2, 3 and FADH, it will undergo oxidative phosphorylation. What is oxidative phosphorylation? In the inner mitochondrial membrane, there are 5 protein complexes which are located, known as electron transport system. I have marked it in black. All these NADH2 and FADH2, they lose their electron, because how they have NADH is formed, it is by accepting electron. They lose their electron to ETS. As a result, this electron will undergo oxidation reduction in the ETS. Finally, they are accepted by the oxygen to form water. And the important thing is, when electron is moving through ETS, there will be release of energy. What to do with the energy now? The energy which is released during the transport of electron is used to move the proton, because NADH is having proton also. FADH is having proton also. So their protons are moved from the matrix, this is matrix, to the space between inner and outer membrane, that is called perichondrial space or intermitochondrial space. So to that, H plus ions are pumped. So what has happened? NADH has lost its electron to the ETS. When electrons are moving, large amount of energy is released. Energy is utilized to pump the proton to the intermembrane space. As a result, concentration of proton increases in the intermembrane space, concentration of proton decreases in the matrix. There will be a electrochemical gradient which is formed. Now H plus ions are they cannot come through membrane directly to the matrix because they are impermeable. Therefore H plus ion which is there, they come to the matrix, back to the matrix from the last complex, that is known as ATPase complex. When H plus ion moves to the ATPase complex, there will be combining of ADP and inorganic phosphate and ATP is prepared. So what has happened? There was release of NADH and FADH2. those got oxidized in the ETS. When there is oxidation, finally there is ATP formation. That is known as oxidative phosphorylation. These three phosphorylation you have to remember, which occurs during photosynthesis as well as respiration. Now let us check how many ATPs are released during oxidative phosphorylation. There will be release of 2 NADH in glycolysis. 1 NADH when it undergoes oxidative phosphorylation releases 3 ATP. So 2 NADH means 6 ATP. Again, when there is conversion of pyruvate to acetyl-CoA, oxidative phosphorylation releases 6 ATP. Again, through TCA cycle or Krebs cycle, 6 NADH is released, 18 ATP is released. FADH2, 2 FADH2, 1 FADH is equal to 2 ATP, 2 FADH2 is equal to 4 ATP. So total 18 ATP, 6 ATP, here 6 ATP, 4 ATPs are released through both glycolysis as well as Krebs cycle. This is about 3 kinds of phosphorylation. Again, let me summarize. Photophosphorylation refers to preparation of ATP by using light. Substrate level phosphorylation refers to directly from substrate phosphate is added to ADP to prepare ATP. Oxidative phosphorylation refers to when NADH and FADH2 undergoes oxidation in the ETS, there will be release of ATP. These are the 3 kinds of ATP. I hope you understood. Thank you.